Okay, so here we have the first panel of the first figure of the paper. In the y-axis, we're seeing 8 of the features that consistently were some of the most important of the 2,300 or so features. In these box plots, so each of these rows, there are about 68 diamonds representing, where each diamond represents one of the different aged mice compared to a 4-month-old mouse. And in the x-axis, we have sort of the rank of that feature in that particular comparison. And what we really are seeing in this figure is that the length features, the features that are associated with transcript length, tend to be the most important of all of these features. So we have best ranked length. So that is the best ranked length feature of all of those tests. And then we have transcript length, we have gene length, and we have coding sequence length. So those are all features that are associated with transcript RNA length. And besides those, there were important features like total transcription factors that were present in the RNA or that were present in a particular RNA's regulatory sequence. So this next feature gives one example of the esophagus tissue for a particular for a 12-month-old, which was the oldest, 24-month-old versus the 4-month-old, which was the oldest mouse in the data set. And what we're seeing is that transcript length, sort of as we're looking at RNAs that are longer and longer, we're seeing this really strong Spearman correlation of a sort of decrease in relative fold change in expression. So you're seeing longer transcripts tend to lose their expression with older age. This third panel of the figure shows the length correlation. So it's similar Spearman correlation that we see in panel B, but for the different aged mice. And so we're seeing generally a negative correlation. So it appears to be a phenomenon that occurs pretty early on in the mouse's life, but it's most significant definitely in the 24-month-old mouse. And if we just looked at all of them in general, we would see that, just by chance, length should be relatively neutral. And then this last example gives so it basically takes all of the RNA in this data set, and it basically bins them in whether they are decreasing in expression or whether they are increasing in expression. And what we can see here is that the transcripts that are decreasing in expression tend to be a lot longer than those that are increasing.